In this tutorial, you will learn the differences between the types of microscopes and the key viewer elements of each scope. The field scope, dissecting microscope, and the compound microscope are optical scopes that use visual light to view objects. The scanning electron microscope, SEM, and the transmission electron microscope, TEM, use electron beams to view images. Using electron beams allows you to view much smaller objects with greater detail. You would use these types of scopes to view microscopic objects. Each type of microscope also differs by the types of objects that can be viewed. The field scope, as its name implies, can be used to study species in their natural habitat, such as a bird's courting behavior or other species' social behaviors. The dissecting and SEM microscopes are used to study the external structures of objects but with increasing degrees of magnification. For example, the dissecting microscope would be used to study the external structures of an insect, and the SEM microscope would be used to study the external shape and structures of a red blood cell. The compound and TEM microscopes are used to view internal structures by preparing thin slices of samples and then looking through the slice. Both microscopes are good for analyzing tissues, cells, and cell structures, but the TEM microscope has the ability to view extremely microscopic objects, even down to the subcellular level. When a species is chosen in the species selector, the projectors will display a set of thumbnail images available for that microscope. Clicking on a thumbnail image will allow you to view the image at full size, as if you were looking at the sample through a microscope. Let's take a look at the different viewer elements. You can use this slider control to adjust the contrast of the image, useful in exposing subtle features of an image. These buttons can zoom and pan the image. The description below the image area displays relevant information and metadata about the image. The image scale shows the approximate size or scale of the image at its current zoom level. Some species, like amoeba or human, will display a video, indicated by the play icon in the thumbnail image. Clicking one of these images will play the video in the image viewer. When a video is displayed, use the controls to play and pause the video or to skim through the duration of the video. This concludes this tutorial on the different types of microscopes and the key viewer elements of each microscope.